Okay, so next up is the fibrocartilage. We studied some of the examples or anatomical areas where the fibrocartilage is present. It is present in, can you recall, where is the fibrocartilage present? Well, one of the important places where fibrocartilage is present is the pubic symphysis, right? The pubic symphysis is one of the areas where the fibrocartilage is present. Another place where it's present is the intervertebral disc, okay? So intervertebral disc also contains the fibrocartilage. Which part of the intervertebral disc do you think contains the fibrocartilage? Well, it is the, is it the nucleus pulposus or the annulus fibrosus? It is the annulus fibrosus, which contains the fibrocartilage. Now, you take a piece of the annulus fibrosus, or you take a piece of the pubic symphysis, you examine it under a microscope with a proper dye. What you see is something like this. So what you see is two different kinds of cells. Firstly, so it contains two different kinds of cells. One of those cells are enclosed in lacunae. So the cells that are enclosed in lacunae would be the chondrocytes. And there's also some fusiform shaped cells as well. And those fusiform shaped, let me just zoom into this image right here. There is cells that are enclosed in lacunae, as you can see right here. And then there's some other cells as well. And these cells are of the connective tissue elements. And these cells that form the connective tissue are called, called what? Fibroblasts, right? So it contains two different kinds of cells. Cells that form the connective tissue and what form the cartilage extracellular matrix. Now, what are the fibroblasts doing there? Well, the answer to this is very simple. The structure of the ECM of fibrocartilage also contains collagen type 1 on top of collagen type 2. So it contains two different kinds of collagen. One of them is collagen type 2, which was present both in hyaline cartilage, type 2, which is present in both hyaline cartilage and elastic cartilage. And on top of that, it also contains collagen type 1, okay? And this type 1 collagen is coming from these fibroblasts that are also present inside this form of cartilage. So it contains two cells that are the fibroblast and the chondrocytes that are in the lacunae. Another feature of fibrocartilage is that I'm going to bullet point this and then bullet point another reason. Another factor of the fibrocartilage is that fibrocartilage does not have any perichondrium, okay? So there's no perichondrium present inside fibrocartilage. So there's no perichondrium, no dense connective tissue containing those MSCs, those fibroblasts-like progenitor cells that are present in the fibrocartilage. Now, because it contains more collagen and some fibroblasts as well, but it contains more collagen, do you think it would stain more pink or more basophilic? But obviously, it would stain more pinkish, right? Because it has more collagen. More collagen means more pink, and that's exactly what happens. Is that in fibrocartilage, you can see almost visible layers of collagen type 1 and densely stained collagen among the different groups of cells. So, for example, in this photo, we see that it contains a very densely stained layers of collagen type 1, and it has these almost isogenous group layers of chondrocytes as well. So, isogenous groups of chondrocytes surrounded by fibroblasts and collagen type 1 and 2. A very similar case in this photo as well. It has these extracellular matrix with collagen fibers that are very, very visible. Okay, that's pretty much it for the fibrocartilage. So it has basically collagen type 2, type 1, and it has no perichondrium. And that's basically the differences that you can see in the histology slide is that it has a very pinkish ECM with layers of collagen, and it has two different kinds of cells, and its chondrocytes are arranged in almost straight isogenous groups. So that was pretty much it for the fibrocartilage. You can review the things later in the neat notes. But here I have some of the a compilation of all of the different cartilages and their histological structures. So immediately we can, or we should be able to see the differences between the three different types of cartilages, because we have studied the different types of cartilages. All right, so let's start with the extracellular matrix. Okay, so hyaline cartilage contains a very clear looking extracellular matrix as compared to elastic cartilage, which has a very it has very visible elastic fibers, elastin, and it also has a very densely stained and variable non-homogeneous ECM. And again, a fibrocartilage over here, what it contains is layers of collagen fibers, and it also contains the chondrocytes that are organized in cell rows. Okay, and we looked at the cell rows over here in this photo that I'm going to go back to. Right here in pink, we have rows of chondrocytes that are arranged in this in the fibrocartilage. However, these are not arranged in rows, but instead are present in their own lacunae in hyaline and elastic cartilage. So those are the differences. And then there's obviously anatomical differences between them, and those are dependent about more about their function and their locations, which you study more in general anatomy. So here's those were the diagrams, and this is the histology slide example. So this is the hyaline cartilage, homogeneous ECM, and not densely stained, and does not contain any fibers. This one is not very densely stained, but it does contain some fibers, collagen fibers that are easily visible. Similarly, elastic cartilage also contains some elastic fibers, and also it has a very densely stained ECM. Now, fibrocartilage contains two different kinds of cells, right? While elastic cartilage and hyaline cartilage only contains one type of cell. So what cells do fibrocartilage contain? Well, fibroblast and chondrocytes. Another thing that fibroblast contains is that it contains two different kinds of collagens, type 1 and type 2, right? While elastic cartilage and hyaline cartilage only contain type 1 collagen. And that's pretty much it for this chapter. So hopefully you guys will be able to understand the differences, the different features, and I have accumulated some neat notes for you guys. I'm going to show you a little bit of them over here. Okay, so here are the neat notes, and let's go over some of the clinical correlates. So firstly, we have the erythritis that we studied earlier as well, is that it is the age-related wear and tear of reticular cartilage. So age-related wear and tear of the cartilage, especially between the articular joints, between the synovial joints. And what happens is that when this cartilage disintegrates, there's wear and tear of it, then the bones instead of the cartilage end up making some of the contact. Yeah, so we studied that the cartilage itself does not contain any sensory nerves, right? But there's nerves that are present on top of the bone. So what happens when bones make contact with each other, especially during shock absorption, and weight bearing? What would happen, do you think? Well, it causes lots and lots of pain, right? So that's basically what erythritis is, and it also results in even inflammation, and inflammation even releases what's called MMPs, or matrix metalloproteases, which are able to even break down further reticular cartilage, and it accelerates the process. Yeah, so this process can be slowed down by giving supplements, right? And we discussed some of the supplements like chondroitin. We said that chondroitin is a major part of the hyaline cartilage, and hyaline cartilage is present inside the articular cartilages, articular joints. And so we can slow this process down by giving chondroitin and glucosamine supplements. Okay, another clinical correlate is the calcification of cartilage. Cartilage can be calcified. We study more about it in the next chapter, but basically, calcification of cartilage results in the formation of bone. And with the progress of age, different cartilages in the bones of human body can be ossified. And these are especially ossified in the costal cartilages. So the bones of the rib ribs, in front of the bones of the ribs there's costal cartilages, right? And those costal cartilages attach the ribs to the sternum, and those costal cartilages are often seen to be calcified in older individuals. So some tumors over here. Remember, we studied in the previous chapter, we were doing the adipose tissue, and we studied that lipoma is the benign tumor, while liposarcoma is the malignant tumor of adipocytes. Similarly, we have chondroma over here and chondrosarcoma over here. So chondroma would be the benign tumor of cartilage, and chondrosarcoma, sarcoma basically stands for cancer. Chondrosarcoma would be the malignant tumor of the cartilage. All right, so we studied some of the basically differences of elastic cartilage, fibrocartilage, and basically formation, growth, and repair. We did the two ways that cartilage can grow and newer chondrocytes can form. One of them was mitosis, right? From pre-existing chondrocytes, mitosis occurs. They form two identical chondrocytes, and this type of growth is called interstitial growth. This interstitial growth is basically happening inside the interstitium, inside the ECM. While appositional growth, which was occurring from the progenitor cells from the perichondrium, and that growth is called the appositional growth. Okay, the repair is very, very slow, or you could say that there's almost no repair, because cartilage is very, very avascular, okay, and there's a lack of mesenchymal stem cells as well. So when there's a lack of mesenchymal stem cells, there's not a huge source of newer cells, and there's also avascularity, then the growth is very, very slow, or almost there's no growth. This is why especially in individuals who have osteoarthritis, who have the wear and tear of articular cartilage, it's often very, very difficult to treat. Okay, here's a table that I absolutely am in love with for this chapter. It contains the differences between all the different types of cartilages. You can see the images later by rewinding this video, but I would like you guys to pause this video and have a read of the differences. It will help you review what we have learned and help you learn as well. So that's pretty much it for the for this video, for this chapter. And next up, we are going to start with the bone. So hopefully I'll see you guys in the next one. Good luck.